Hello everyone, today our topic of discussion is the epithelial tissue. Now the epithelial tissue is classified into two groups. One is the simple epithelium and one is the compound epithelium. Now the simple epithelium is categorized into four parts. One is the squamous, which is also known as the pavement epithelium, the columnar, the ciliated and the pseudostratified simple epithelium. Okay? And the compound epithelium is divided into stratified and transitional epithelium. Now the stratified epithelium is further subcategorized into stratified squamous and stratified columnar epithelium. But now the stratified squamous epithelium will again be differentiated on the basis of keratin content into keratinized and non-keratinized epithelium. Okay, now we are going to discuss about the simple epithelium. Now the simple epithelium is a single layer of epithelial cells, okay, resting on a basement membrane. So they are generally like this. Now they contain a single layer of cells, okay. Now they rest on a basement membrane and is present on the areas where absorption is more but wear and tear is less. Simple epithelium consists of the following types: the pavement or simple squamous, columnar with or without cilia, pseudo stratified. Okay, now let's discuss about the pavement or the squamous epithelium. Okay. Now the pavement of the squamous epithelium are flat cells with irregular outline and fit together by their edges to form a continuous thin membrane. See, this is how they are grouped with each other. Their boundaries are irregular. Okay. Now they contain the nucleus. So these are the nuclei. Okay. Now the cytoplasm of the cells spread like a film and the flattened nuclei are visible. So these nuclei are generally flattened. Okay. Don't make it a sphere. Actually they look a bit flattened. So this epithelium is adapted to perform a dialyzing or filtering function. Now distribution. Now when we come to distribution, the pavement epithelium is found in the linings of lung alveoli. Okay, it is found in the linings of lung alveoli. Number 2, the inner surface of the serous cavity, inner lining of heart, blood vessels and lymph vessels. And also in the loops of Henle of the nephron. So this is how the squamous epithelium is found in different parts in our body. Okay, now to talk about the linings of the lung alveoli, it is endodermal in origin, whereas the rest of the pavement epithelium is mesodermal in development. So this is one important thing to be noted, that the lining of lung alveoli is generally endodermal in origin. Okay, whereas the rest of the pavement epithelium is mesodermal. Fine, I hope it's visible. This one, the lining of lung alveoli is endodermal and the rest of it is mesodermal in origin. Now the term endothelium, okay, when we talk about the term endothelium, now the endothelium is applied to the cells which line the blood and lymph vessels. Okay, now the lining cells of the serous cavity are known as mesothelium. This is for the lining of the serous cavity. Okay, and this one are the lining of blood and lymph vessels. Fine. Now this was our number 1. Now number 2, we come to the columnar epithelium. Okay, so now let's discuss about the columnar epithelium. Now we will discuss about the columnar epithelium. Now the columnar epithelium are generally cylindrical in shape. Okay, now they rest on a basement membrane and the nuclei are generally oval, placed centrally or towards the basement membrane. Okay, so this was how generally the columnar epithelium is oriented. Now there are a subcategory of columnar epithelium which is known as the cuboidal epithelium or the cuboidal cell. Okay, now what is the cuboidal cells? Cuboidal cells is generally short columnar epithelium. They are short columnar epithelium. Now some of the columnar epithelium are specialized by possessing brush border or cilia. In addition, there is one important cellular structure, okay, which is called the goblet cells. Okay. Now the goblet cells belong to a special category of columnar cells. Okay. Now we are going to discuss about first let's discuss about the structure of the columnar epithelium. Okay. So generally the columnar epithelium is like this. So these are column-like cylindrical cells. Okay, and their nuclei lie towards the basement membrane, and they may also bear cilia on their free surface above, which will make them the ciliated columnar epithelium. And as far as cuboidal epithelium is concerned, cuboidal epithelium is generally cube-like. Okay, it is generally cube-like. They also have a basement membrane, and nuclei is more or less centrally placed. Okay, so this is the cuboidal and this is the columnar. Okay, bear cilia, so it may be also termed as ciliated columnar epithelium. Now distribution. Now the columnar epithelium is distributed into some important areas in our body. Now the first one is the gastrointestinal tract. Now the distribution of the columnar epithelium, it is generally found in the gastrointestinal tract, okay, and its glands. The greater part of male urethra or the vas deferens. Okay, the gastrointestinal tract and its glands, greater part of male urethra, vas deferens, and the follicles of prostate, vulvourethral, and greater vestibular glands. Fine. So let's recap about the distribution. First, the pavement or the squamous epithelium, it was found in the linings of lung alveoli, the inner surface of serous cavity, the inner lining of heart, blood vessel and lymph vessels, loop of Henle of nephron. And the columnar epithelium is found in the gastrointestinal tract and its glands, greater part of male urethra, vas deferens, follicles of prostate, vulvourethral and greater vestibular glands. Now let's discuss about the goblet cells, which is considered as a special type of the columnar cells itself. Okay, the goblet cells are modified. Okay, they are modified columnar cells. They contain mucinogen granules. Okay. They contain one important set of granules called the mucinogen granules, which is found between the nucleus and the free surface of the cell. Now the secretion presses the nucleus towards the deep part until the contents are discharged from the free surface of the cell. Now the mucus secretion of goblet cells is manufactured by the mucus secretion is manufactured by the endoplasmic reticulum, the rough part. Okay, so the mucus secretion is regulated by this cell organelle. Now it is stored and excreted by smooth surface vesicles of the Golgi apparatus. Now coming to the distribution of the goblet cells. Now the goblet cells is distributed in the gastrointestinal tract and in the respiratory system. Okay, so this was about the goblet cells. Understood? Now let's discuss about the brush border cells. The brush border cells. Now the brush borders are some of the tall columnar cells which exhibit vertical striations. They exhibit vertical striations known as the brush border at the free surface of cells. Okay. Now when you see them using electron microscopy, it shows that the brush borders are formed by numerous microvilli. Okay. Now which are cylindrical projections of the cytoplasm of the cell, somewhat like this. Okay. So these are the microvilli. Now they are cylindrical projection of the cytoplasm of the cell. They are covered by a cell membrane. Okay. Now the brush borders are rich in one important enzyme known as the alkaline phosphatase. Okay. They are rich in this enzyme. Alright. Now they are found in, when we come to distribution, they are found in the small and large intestines. Okay, and in the PCT of nephrons. Okay, a single cell of the intestinal villi, okay, presenting brush borders may have 3000 microvilli. Now this microvilli helps in the absorption of the chemical substances and sugars from our body. Okay, so we have discussed about the goblet cells, brush border cells, which were a special type of columnar epithelium. Now after discussing about the special columnar cells, that was the goblet cells, the brush border cells, and the ciliated epithelium, which is left to be discussed. Okay, the goblet cell is discussed, and then we studied about the brush borders, and now we are going to study about the ciliated epithelium. Now the ciliated epithelium, it consists of simple columnar cells. Okay, now it is also a special kind of columnar cell. Okay, generally on the basis of cilia, it is differentiated. Okay, now it consists of the special columnar cells which are provided with filamentous threads known as cilia. Okay, this thread like filamentous threads. Okay, now the cilia may be motile or non-motile. Okay, they may be motile or non-motile. Fine. Now they be motile or non-motile. Now the cilium is covered by the cell membrane and contains within the matrix 2 microtubules in the center and 9 paired microtubules at the periphery. Okay, so there will be 2 microtubules. At center and 9 paired microtubules at periphery. Okay. Now the peripheral paired microtubules are arranged in figure of 8 fashion. Okay. They are generally arranged like this way. Okay. So now drawing the whole microtubule, just imagine that the 9 pairs are arranged in the form of a circle. Okay. Now one tubule of each pair bears tangentially oriented arms, and such tubules are referred to as the subfibers A. Okay. So they are referred to as the subfibers A. Generally what happens is, see, this is the microtubule. Okay. So this is the whole, the cross section of the cilia. So these are the central microtubules which is here, this tube, and here are 8, pairs of 9 microtubules. 3, 4, 5, 6, 7, 8, 9, suppose it's here. Okay. Now one of these microtubule will bear a tangentially oriented arm, and this tubule will be referred to as the subfiber A, and the other tubule of the pair which do not possess arms are called subfiber B. Okay, subfiber A and subfiber B. Now the arms are pointed towards the adjacent paired tubules in clockwise direction. Okay. Now when seen from the base to the tip of the cilium, they will be in a clockwise fashion. Okay. When seen from base to the tip of cilium. Okay. Now let's discuss about the cilia in a more detailed way. Okay. Now the cilia of the animal kingdom generally consist of the 9+2 arrangement. Okay. 9+2 tubular arrangement. Alright. Now cytochemically, the microtubules are composed of actin contractile proteins. Each cilium consists of a tip, a shaft, and a base. Now located close to the free surface of the cytoplasm, there are attachment of basal nerves. Okay. Now filamentous rootlets on the basal nerves extend further deeply into the cytoplasm for better anchorage. Okay. Suppose this is the cell membrane and this is the cilium. Okay. So they are attached through anchoring proteins for better anchorage. Now there is a category of stereocilia which are devoid of microtubules and resemble enlarged microvilli. Okay, they are the stereocilia. Now the ciliary bead consists of 2 successive phases. Okay, the ciliary beads. One is the effective stroke. Okay, and the other one is the recovery stroke. Now what is the effective stroke? Now the effective stroke can be represented in this way. Suppose this is a tubule, and when it gets directed in this way while contraction, okay, in effective stroke the cilium bends at the base. Okay, it bends near the base, but the shaft remains stiff. Now during recovery stroke, what happens is the wave of bending of the cilium extends from the base to the tip until it regains the original position. Okay, so what happens is now it will retract its way and again come to its normal position. Okay, it's like generally what happens during a vibration. Alright. Now such waves of ciliary beads progress in the same direction and affect the adjacent cells one after another. Alright. Now the distribution. Now we'll talk about the distribution on the basis of motility of cilia. Now the motile cilia, okay, is found in the respiratory tract, the auditory tube, the tympanic cavity, the uterine tube, the uterine cavity, okay, before puberty. This must be noted. Now the non-motile cilia is found in the ventricles of the brain, okay, the central canal of spinal cord, the epithelial cells of epididymis, the ampullary crest and macula of internal ear. Now let's discuss about the function of the cilia. Now cilia weights rhythmically and help in the transport of cells or other foreign bodies. They protect the respiratory tract by eliminating the bacteria and foreign particles which are trapped by the sticky mucous membrane. Okay. So this was about the cilia, okay, the effective stroke, the recovery stroke. Then we discussed about the distribution on the basis of motility of cilia into motile cilia and non-motile cilia. Now the cleansing action of cilia is done by exciting the cough reflex or sneezing reflex while you sneeze, okay. Now the respiratory tract acts as the mucociliary barrier, okay, to the bacteria and helps to prevent from getting inside our body. Now the ciliary bead is affected by temperature, okay, and it may be affected by the temperature, the pH and osmotic pressure as well of the surrounding medium and various chemical substances. Now there is one important syndrome which you may encounter in any patient, which is known as the immobile cilia syndrome. Now what happens in this case? See, the immobile cilia syndrome, what happens is it takes place due to defects in genes coding for ciliary proteins, okay. Now the cilia are sensory in function, okay, they're generally sensory in function in the ampullary crest of the internal ear. Now the functions of stereocilia are still not known, okay, possibly to increase the absorptive surface of the mucous membrane. Now just as a recap, what we have discussed till now, first we discussed about a pavement or the squamous epithelium, then we discussed about the columnar epithelium. We learned about the different special categories of the columnar epithelium. Next we learned in detail about the cilia, okay, and we learned about the different movements of cilia, which was effective stroke, recovery stroke. We learned about its motility and distributed them respectively on the basis of motility into motile cilia and non-motile cilia, okay. Now we are left with the last of the simple epithelium, last topic of the simple epithelium, which is the pseudostratified epithelium. Now in some situations what happens is the ciliated columnar, okay, the ciliated columnar cells, similar like the ciliated columnar cells are of different heights, okay, so they may be like this, okay, and like this, and maybe like this, and maybe like this, yeah, all of them we are cilia, more or less everyone has a cilia. Now what happens is they are on a different heights, okay. Now but they rest on a common basement membrane. All cells do not reach the surface and the nuclei are placed at different levels, okay. Some of its nuclei may be here, some may have here, okay. Now all the cells do not reach their surface, and so what happens is their nuclei are at different levels, resembling false stratifications. What happens is they may look like as if they are falsely stratified, like this. Suppose his nuclei is here, okay, but they consist a single layer. Okay, but there may be modificational changes which would make you think like that they consist of different stratas. Okay, but they resemble it totally in a fake fashion because it's a false stratification. Hence, this type is known as the pseudo-stratified columnar ciliated epithelium. Now, where is it found? It's found in the mucous membrane. Okay, the mucous membrane of nasal cavity, in the trachea, in the bronchi. Okay, mucous membrane of nasal cavity, trachea, and the bronchi. The generally found here. Okay, so I hope you are seeing it clearly. Now, the shorter cells that fail to reach, okay, these are the shorter cells. Now, these shorter cells who fail to reach the surface probably serve as precursor cells to replace these longer cells, okay, which are lost from the membrane in subsequent period of time. So this was pretty much about the simple epithelium which we discussed. Okay, we discussed about the classification of simple epithelium. We discussed about the classification of epithelium in general. Okay, we discussed about the types of the columnar epithelium. We learned about the cuboidal epithelium, the goblet cells, the brush border cells, the ciliated epithelium. Next, we learned about the cilia in detail, its distribution on the basis of motility. Next, we learned about the pseudo-stratified epithelium and where is it found. So this was pretty much about the simple epithelium. We're part of this video, okay, where we will be discussing about the compound epithelium. Okay, so hope you understood well. Thanks for watching.